Okay, good afternoon. Um, we will talk about gene therapy. I think you already understand what is meant by gene therapy. Typically, why this is important, we will discuss in a short while, or the potential of it to understand the potential of it. You need to think of issues like this. If there is a defect in a person, and that defect is because if that defect is because of a gene, lack of a gene, or improper regulation of a gene, and then the best way is to, I mean, some problem with the gene, and it causes some symptoms or some changes in phenotypes which are usually not good for the individual, right? In that case, what should you do? Should you try to treat the symptoms, or should you try to rectify the gene or the gene product, right? There are several ways of approach to the question, something like this. Say it is the same as if there is a pathogen, a bacterium that is causing disease. Should you treat the symptoms of it, or should you get rid of the pathogen? Okay, so while both are good, both are important, sometimes treating the cause itself might be difficult, so we treat the symptoms and try to mitigate through the situation. And one of the approach in genetic disorders, at least, is to try to rectify it. The problem is, if the person has a disease and that person is going to has the genetic defect, and it is going to propagate to his children or her children, and the person, if the person gets treated, he will be fine, but again the same issue might be having with the progeny and generations together. If it costs about, say, 1,000 rupees, okay, 1,000 rupees, then imagine how much money we have to spend for generations after generations, right? So there are approaches to rectify the gene. You can also have it, you can think of it as rectifying it in somatoplasm, or somatoplasm, that is the tissue, that is what the tissue that is in which the gene has a role to play. We're only dealing with the tissue. Or the other one is germline, germ plus gene therapy at the level of germline, so that if we treat one person, we don't have to deal with the same issue for the progeny. Okay, but let's just try to see what we have. There are many genetical diseases. Some of these are not necessarily genetical, some of them are environmental issues or developmental issues. But typically, a genetic disorder is an abnormal condition because of the inherited genes or chromosomes. So it comes from parents to children, and so on and so on. So we need to first address the individual who is suffering, and only then we can try to think of how to uh, how to take this or rectify this in the species level. This one is at individual level. If you're trying to, as I was showing in the before, if you're trying to address only here, we are treating it at the individual level. If you are trying to go for the germ line, then we are actually trying to think in the species level. Okay, so there are a multitude of um, genetic disorders because of, it could be a gain of function disorder or a loss of function, typically a loss of function, and then which would lead to developmental issues and many problems. In humans, the great thing about humans is that we do care even though they have a problem or disease, we do take care of them. I have the problem with Indian scenario is in some cases if somebody is born with a an abnormality like an extended nose or something, then you start praying to them. Those are several articles also shown that they're available on on internet. You can check it out. But these are something we need to address, and these are some of the very disturbing uh, type of skin diseases where they do not have, they fail to grow while the body grows. Some of them fail to produce sebum, and so they have to be continuously moisturized, which otherwise would crack, like we have seen in the picture. I'm just closing it so you don't have to see it for a longer time. So genetic disorders do move from, I mean, are transmitted from one generation to another generation. And of course, if there is no natural selection, right? Natural selection is reduced in our in humans because we are not Indian. Sorry, not Indian. In general, human population is not about biological fitness anymore. Biological fitness, when I say that, it means that the person has all the genes of good health, for a good health, for a happy and long health. So we are not, there is no more individual to individual direct physical fight. There is no physical fight to determine who is fit and who is not fit. The environment, the natural, the environment will impose fitness. We don't have that also. We use science and technology to mitigate the natural selection, the environment posing an selection on us. What we are, we don't have any more physical conflict, so it's not like the strong might is right, that is no longer working, or the way might is different now. Who is having more money? Who is having more power? Power in the sense social power. Who is having a tool? It could be a knife, it could be a gun. How sophisticated is the tool? So fitness is now dependent on these things. Power is about social status. It is about how many followers the person has, or if you just whistle and how many people will come. Like in movies. So it has nothing to do with how strong you are, how much muscles you have, what your skills are. Your skills now, our skills now should be about money matters, power matters, and about weapons and other things. If there is a conflict, I'm telling you. And upon it, this is too huge actually, because it's about how you form a group, how you form a band of people, how you bond with several types of people. You can instill a kind of emotion or a kind of belief systems that something happens, then all will group up, and then there is a tremendous power. So what I'm trying to imply is that the natural selection is no longer, is very little acting on humans actually. It has to be, somebody has to be really really bad to not be able to survive and reproduce. So ours now is more of social fitness. So there should be a, there is there is an improper balance between biological fitness and social fitness. Social fitness doesn't require you to be the smartest person, doesn't require you to be the strongest person. Say for example, a parent rich parents, even if the child is stupid or unhealthy or anything, it doesn't matter. The person will give the money to the child. It will come. That's what we all do. That's how I get from my parents. That's how my children will inherit from me. That's what we all do. So This is a kind of fitness. If you have to see money as money can be seen as a fitness. That fitness is not coming through genes, it is coming outside. It's a biological. And if the child or somebody who is even unhealthy still has a chance to get a chance of reproductive chances are bright. You can think of it like this: a very healthy person, uh, poor or does not have any social influence, and a crippled person uh, who may have, he is rich, may have a chance of reproduction compared to this person. Desirability, right? Desirability quotient is different. So the the point I'm trying to put in is to see clearly through, see the nature of human society and how and what kind of implications might be there in the future. There is nothing to be political about it, or trying to be politically correct is not right. When we have to see the truth, we have to see the truth. That is what is happening. I'm not saying if it is right or wrong. What I'm saying is that is what is happening. So what should be done is to have a way of rectifying these genetic disorders. You will be surprised to know, I think more than 4,500 genetic defects are available in the in the current human population. I don't think any other species has like that. No other species would have as many genetic defects as humans have. The problem could be our excessive dependence on social fitness, excessive dependence on science and technology to mitigate the environmental factors. It could also be that our choice of selection is, there is kind of sexual selection is also biased, right, towards social fitness. And we all probably have one or two genetic defects, or genetical predisposition is also there. So we should try to ensure in the future, for the sake of our future human species, what we should do is to try to address these genetic disorders, how to reduce them, because we don't want people to be suffering. We don't want to spend so much money just for the treating the ill, because the only thing that is going to happen in the future is more and more of uh, problems will be coming up, and healthcare industry will be the biggest industry anywhere. So the point how to cure a gene is to, if there is if a disease is caused because of a defect in the gene, there are, can we replace, that is one thing, and can we repair, right? Just repair the small mutation that is there. In some cases we may have to replace the whole gene if there are things. Either way, it is still a genetic manipulation, or I should actually say genetic correction, right? Trying to restore it to a um, normal genotype, or wild type genotype. I'm not talking about enhancing the human population or giving characteristics that this person may not be, or a no human is normally having. Something like putting a frog or some kind of gene into a human to enhance, so that he or she will be able to jump like a frog and then win all the medals in the Olympics. I'm not talking about enhancing the species. I am talking about correcting the species. Okay. So there are multiple ways of how things are done, and I will just skip this. And this is what I was talking about: somatic therapy and germline therapy. Somatic therapy is treating the individual. Germline therapy is treating the whole lineage or the future lineages to come. So this is somatic one is typically not inheritable, whereas in germline we are hoping that it is going to be heritable. And the approaches have different ways which tissue you are working on. Usually if it is germline, we are working on the ovary or testis, or actually more specifically spermatogonial stem cells or oogonial stem cells and so on. There are two approaches. One is called as ex vivo gene therapy, and the other one is called as in vivo gene therapy. We will talk about ex vivo gene therapy because we are much more, we know it. We have already finished it in a way that is almost similar to that of engineered embryonic stem cells. Engineered embryonic stem cell approach of transgenic animals. It's not, the whole thing is not exactly the same, but it is almost similar to that. So we take cells, we take the cells from the patient, and we would have made a therapeutic copy or gene that we want to change or restore. And again, you have to remember about the gene construct that we have done when we have studied about transgenic animals. I described what are the components important for gene targeting, what are the components important for homologous recombination, positive selection, negative selection, and so on. The same things apply here also. As a part of gene therapy here, we are actually talking much more general overview. So the gene construct with the correct gene is taken and inserted into viral vectors, and we would have taken the target cells and we would grow them in the culture and allow the infection of this phage to the cells, and then we would have to select the cells that are transfected, and we should have also characterized where did it integrate, is there any other change, if if the cell is fine or not. One of the problems with anytime we are trying to do with gene therapy or manipulating the genes is transformation. Okay, anytime you hear the word transformation, when it is with eukaryotic cells or animal cells, it would mean something becoming cancerous. In bacteria, if you're listening to something transforming, it means it has acquired a DNA from naked DNA from outside. So keep this terminology clear. Here I'm telling, anytime we make a genetic modification, there is always a risk of transformation, that is becoming cancerous. So this part becomes a big issue, where how you select and characterization to characterize, and that the cell is fine and everything else. And then we introduce the cells into the body so that they would go and function as if they are normal with a corrected gene. We call this ex vivo because all the manipulation or the correction of the gene is done outside the body, and that's why it is referred to as ex vivo gene therapy. Okay. There are multiple things here. One has to understand that there are problems with what kind of cells are taken. Not every cell can just become corrected. We might have to sometimes go for induced pluripotent stem cells, then derive and make the correction in the induced pluripotent stem cells, then derive whatever the cell type that we want, and then put it into the patient. That would be the actual version in the future. Everybody in the future may have, as soon as they're born or so, they might take a few cells from the maybe embryo stage or so on, and try to store their make them induce pluripotent and store them for future. Anytime the person needs, they can derive whatever cell type the person needs, and they might be putting it. That will be the highest form of community regenerative medicine. Okay. So in vivo gene therapy is much more non-specific. Ex vivo has critical details about this is what we will do and so on. In vivo gene therapy is little bit troublesome for several reasons, but it is the easiest one. We will try to have a look at that. First thing, we will have a DNA, the same thing as therapeutic. The viruses, the gene of interest along with the vector is directly given into the body and it to the target cells, and that the released DNA is hopefully it will get integrated into the genome, replacing the faulty gene, and then it gives rise to a functional tissue also. We call this in vivo because we are the gene therapy or the correction of the gene is happening within the body, right? And hopefully that is why it is very simple to as a concept it is simple, and there are problems however. If we should know the choice of the vector or the choice of the virus that we are choosing should depend on that virus should have specificity. Excuse me. Sorry. Yeah, the virus should be able to infect that particular tissue. Only then it will make sense. Otherwise, it doesn't make as much sense, isn't it? Because if you are putting a virus and the tissue we want to, sorry again, sorry, yeah, where was I? Yes. So the specificity of the vector, the virus that is chosen should be to the target cells. And if you are putting in DNA, again, sorry, yes, yeah, that's about in vivo gene therapy. The good thing is it's very simple, and if it works, I think this should be the best way to do stuff because it's inexpensive. You don't need much to do. All you have to do in vitro is only this much. Whereas in the ex vivo, everything is done in the outside the body, and then you put these cells there. In vivo, in vivo type, the in vitro techniques that you are employing or manipulation is very little, but it's highly inefficient. I hope you see that when we put the vector, it should not, it should reach the cell, it should get deliver the DNA, and the delivered DNA should get incorporated. And if you have, say, 1,000 cells in a tissue, maybe only 10 cells would actually be transfected. That is on a good day. And then if you put in these phages and other things into the body, there might be some immune reaction, the removing of immunocomponents or immune cells might remove the DNA. And there is also another risk. If this tissue goes and activates, delivers the DNA to some other tissue, which we don't want this gene to be expressed in that tissue, it's abnormal in that case, and that is a problem too. So this is kind of non-specific in a way, because the probability of other integrations and complications is much higher. And then there is, anytime, as I said before in the previous slide, this transformation or cancer formation is always a problem with gene therapy. But in ex vivo, I think it's kind of okay, because you could have tested it if it is transforming or not before you before we incorporate the final engineered tissues into the body. But here in vivo gene therapy, it sounds it would be a miracle if it works out without a complication. Yes, but these are the approaches. As you can see, why in vivo gene therapy is appealing, because of its simplicity, and it is inexpensive, and so on. So both have their ups and downs. Yeah, these are the same things, basically talking about little more in little bit details about whether embryonic stem cells are taken or from SCNT, or embryonic stem cell lines from HLA banks, or you can also add induced pluripotent stem cells and then do the differentiation and the gene therapy and then put it into the person. Or you can also use adult stem cells, by the way. Adult stem cells are much better because they are probably easy to take in, obtain. But if this is a patient already having trouble with that particular organ, and you're taking more out of that organ, then it's going to cause a problem. So that is when we might have to go to embryonic or induced pluripotent stem cells. This side is the direct delivery. It is called as it is the in vivo type, and cell-based delivery is mostly the ex vivo. There is another way of saying things, and this is one of the problems. Some of the things that we need to remember about the target cells. If you are giving, if you are trying to do in vivo method, how do we choose tissue type or organ type, or should we give it systemic? Okay, that again depends on the nature of the correction one is trying to do, but it's always important that we know about the biology of what is happening, what kind of defect we are trying to do, and then act upon based on that. Yeah, I think I'll stop here. But before going, I want to also remind you about these things. Recombinant and you know about the vaccines, right? DNA vaccines and recombinant live recombinant vaccine. What is a vaccine? You have an antigen or a bacterial organism. I'll just use 5 minutes of probably introduction, and then I will stop so that I'll get some breather. Okay, so if there is an organism, right, and a bacterium infects, it causes problems to the mouse for a few days until the immune system activates. Immune system activates and kills the pathogen. And then the antibodies or the memory of the infection is stored in immunity has a memory. So anytime next time the infection happens, the antibodies are readily produced immediately, and that is killed off. It's a kind of miracle for in nature. And also you'll see the similar kind of thing that's happening in when we start reading about CRISPR. So the same principle is induced. The vaccination is about here in the first exposure. There is a problem, right? Some of them, some of the individuals actually may die, or if the pathogen is too strong, they might cause death. So what we do now is we already prime the body with an antigen. We don't give the whole organism. We just give a antigen that is a part of the uh, bacterium or any of the pathogen. So the antibodies are raised against it, and if the real pathogen ever was to come inside, it will be attacked and killed by the immune system. That's the basic concept of vaccination. It is like priming the body or create priming the immune system to already generate and remember the epitopes of a pathogen, so that if the pathogen was to attack an individual, the immune system will act before the pathogen cause death. So recombinant DNA technology is useful in two of those. One is live recombinant vaccines and DNA vaccines. I will discuss these in the next class. Uh, I'm just having this sneeze issues. So, any questions? Sorry. Any questions about anything? Assignments or any of the things? So, in this syllabus, what I have left is recombinant vaccines. I will in the next class or so I will discuss about these vaccines, and then I will also want to talk about detection and identification of pathogens. It is just a revision of some of the techniques that we already finished. Okay, it's not going to be a big deal. Something like RT-PCR, for example, and Western blot, and so on. And maybe we'll these are the same things, trying to use PCR based techniques to identify if there is a genetic disorder or not. And you can use chromosome walking and other things also to find out if there is any chromosomal aberration and so on. So those these are all applications now. We already have studied enough of the techniques. So what we will it is like a revision of what we are supposed to do in these cases. And then gene therapy is what we have discussed today. So in the next class we will discuss about vaccination, and this is about diagnosis, and the other one is about human disease, a few of them. And then we need to move on to two important things. First one, I mean, it's about CRISPR, the biology of it, the potential of it, and then we will also try to have a chapter or a paper on screening for HIV, I think. So we will do the host factors involved in HIV. We will discuss those in the next class. Any questions? If no questions, we will take a break. Okay. We'll meet in the next class. Yes. Take care. Bye bye. Thank you.